everyone, my name is Haley, and this video is going to be all about how I used CRISPR-Cas9 to engineer the bacteria E. coli. The goal of this experiment is to genetically alter the E. coli bacteria so that it can survive on streptomycin, which is an antibiotic that would normally prevent the growth of the E. coli. The goal is to make a specific mutation in the ribosomal subunit protein, or more specifically the RPSL S12 gene of the E. coli, preventing streptomycin from binding and thus allowing protein production. We do this by swapping two codons, which are essentially free nucleotides that code for a specific protein, glycine and threonine, or AA and ACC. And by creating this swap, we're allowing the E. coli to gain antibiotic resistance against streptomycin. And if this experiment is a success, we should see a colony of E. coli growing on our plates. So without further ado, let's get started. So to start, I added 150 milliliters of water into my glass bottle containing my LB agar mixture. I then made sure to shake that vigorously to make sure all the particles were dissolved into the water. I then put that in the microwave for 30-second intervals just to make sure there were no more solids in the mixture. And while you're doing this, you want to make sure that you're not screwing the lid on top, but just putting it on top so that it covers the bottle. After waiting about 20 to 30 minutes for the bottle to cool, and while the bottle is still warm, I'm just going to pour the mixture onto 7 of the LB agar plates. You also want to make sure that you're not pouring the LB agar plate to the top, but just covering it so that it covers the bottom of the plate. I then let my plates cool for about an hour before I flip them upside down to let the condensation evaporate, and I store them in the fridge overnight for the next part of the experiment. So before we move on, we actually have to activate our DNA tubes. We do this by adding 10 microliters of sterile water into each of the tubes, and the result should be a pale opaque liquid. In order for us to grow the bacteria, we have to be able to access it. So we have to add 100 microliters of sterile water into the bacteria tube. Then shaking it for about a minute to ensure the bacteria is dissolved, we then pipette all the mixture onto the LB agar plates. Then using the inoculation loop, we want to gently spread that bacteria so that it can grow evenly onto the plates. And after about a week, my plates were completely filled with bacteria. Since both DNA and the cell wall are negatively charged, they will naturally try to repel each other. This is why we have to add something called a bacterial transformation mix into our E. coli bacteria. The transformation mix contains polyethylene glycol and calcium chloride to help neutralize the negative charge of the DNA and make it more likely for the DNA to enter the cell. Using an inoculation loop, I'm gently scraping off some bacteria and adding it into the transformation mixture, and I keep doing this until there are no more chunks in sight. The fourth step is making our DNA transformation mixture. We do this by adding 3 of our DNA tubes that contain our Cas9 plasmid, our guide RNA plasmid, and our template DNA. Plasmids are essentially a support vehicle for our guide RNA and our Cas9 that help carry them into the nucleus. The plasmid itself is a circular piece of DNA that can replicate independently once inside the nucleus. Now, you're probably kind of lost while watching this, but don't worry, let's break it down. The CRISPR system contains 3 important components that make the genetic mutation in E. coli work: the template DNA, the guide RNA, and the Cas9. The guide RNA does exactly what it sounds like. It directs the Cas9 enzyme where to go in the genome and where to make that specific cut. The Cas9 then cleaves onto the guide RNA and the target gene and makes a double-stranded break. And finally, our template DNA. So once the Cas9 makes the cut, the cell's natural response would be to repair that system through a process called homologous recombination. So if we flood the cell with a DNA template that contains a single base mutation that codes for threonine, we can then prevent the streptomycin from binding and disabling the ribosome, thus allowing the bacteria to grow in the streptomycin. Let's imagine that this diagram is happening inside of our bacteria's cells. The big green blob is our Cas9 enzyme that's cleaving onto the single-guided RNA in our target region, which is the blue strands of DNA. Our single-stranded RNA is the yellow molecule, and finally, the tiny white speck at the bottom is our template DNA, where we want to introduce the mutation to. Moving on to the actual experiment, I then pipetted 10 microliters of each of the DNA tubes into my competent cell mixture. I then left the mixture to incubate in the fridge for about 30 minutes. After it's done incubating, I put it in a jar of warm water that was around 42 degrees warm. The reason why we're doing this is because the plasmid DNA has to go inside of the cell, and to uptake it, we have to create these temporary pores inside the cell membrane so that the plasmid DNA can go inside. And we're almost done. We only have a couple of steps left in this experiment, but now all we have to do is add 1.5 milliliters of room temperature water into a new LB media microcentrifuge tube, which is the same material that we grew our bacteria E. coli on. The reason we're doing this is because it helps grow the E. coli because it's undergoing a lot of stress while it's performing transformation. Next, using the same media that we just created, I'm going to pipette 250 microliters of that into our composite cell mixture that contains our plasmids and DNA. Finally, we incubate our tube for around 12 hours at room temperature so that CRISPR can do its engineering. After 12 hours, the DNA transformation should be finished, so now we can pipette all of that mixture onto a new LB strep plate that we just took out of the refrigerator. Then taking an inoculation loop, we want to gently spread that bacteria so that it can grow evenly. And before we put the lid on, we have to let it sit out for 10 minutes so that the bacteria could cool. After a couple of days, I started to see the bacteria begin to form, which meant that we successfully genetically engineered E. coli to have antibiotic resistance against streptomycin using CRISPR-Cas9. So to recap, I actually wrote an article detailing this entire process that you can check out below. And overall, this technique that we just performed has the potential to revolutionize things like disease treatment and antibiotic resistance. By modifying specific genes, we can potentially find cures for diseases that were once untreatable, like Parkinson's disease, Alzheimer's, cancer. There are so much things that we can explore with this technology, so I encourage you to keep learning and keep exploring. Thank you so much for watching, and always remember to stay curious.